everybody should be somewhat familiar with the heart, and that the fact, we talked about the structure of the heart, the fact that it's a muscle. Those muscles are constantly firing. We know that to be true because your heart keeps beating all the time. But they are specialized in that uh, cardiac muscle fiber, is striated, but it is involuntary. You don't have to think for your heart to beat. You don't have to consciously be in thought for your heart to beat, but there is a special mechanism in place, and that's where we talk about the electrophysiology of the heart, a special mechanism in place that deals with the beating of the heart. So we're going to have a little review first, and then we get into it. So the heart does not require conscious intervention. You don't have to consciously think, heart, please beat for me. You don't have to do that. That's why your heart is beating even when you're asleep. That's very important to remember. The heart the cardiac muscle exhibits autorhythmicity, which is basically it sets its own rhythm, right? So it has this ability. We know the lambda, the do 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 the do 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 do. The beating of the heart. It sets its own rhythm. Right? So without outside input, it can set its own rhythm, which is pretty amazing. And it continues to beat and beat and beat for as long as you're alive. Unless if it stops beating, like I said before, that becomes very problematic very quickly. Between your heart not beating or not breathing, those two things can affect your lifespan drastically. Okay, so that's why cardiopulmonary resuscitation, CPR, is so important. And we're going to talk about the pulmonary system, the lungs and breathing in much more detail when we get to that chapter. All right, but let's continue with the electrophysiology of the heart. So we have these cardiac muscle fibers. We have this ability the heart has the ability to set its own rhythm. The cardiac muscle cells contract in response to action potentials, just like other muscle fibers do. But unlike skeletal muscle fibers and smooth muscle, most cardiac muscle cells do not require stimulation from the nervous system, right? Your brain doesn't have to tell your heart to beat. It's coordinated by these pacemaker cells. We're going to talk about these pacemaker cells, the SA node, the AV nodes. We're going to talk about that in this section. And they create this rhythmic, spontaneous action potential. Right, so let's go into some of the histology. Hopefully you can hear me even if you don't see my face. The cardiac muscle cells, like skeletal muscle fibers, have striations. We know it's striated, alternating light and dark bands. The actin and myosin is very organized. We know that it's a similar structure to cardiac muscle, has a sliding filament mechanism of contraction, but these cardiac muscle cells are not as long as your skeletal muscle fibers, and so you have they're shorter and wider. They contain a bunch of myoglobin, which the protein that carries oxygen, and they have a bunch of mitochondria, which makes sense. These cardiac muscle fibers need to be able to need a lot of energy, right? They need a lot of energy. They're constantly beating versus a lot of your other muscles, your skeletal muscles, your biceps, your triceps, your quadriceps, your vastus medialis, all these muscles get to rest, whereas your cardiac muscles are constantly beating. So they need a lot of energy, a lot of mitochondria. You'll see this structure called the intercalated discs that join adjacent cardiac muscle cells. That's going to be very important. Cardiac muscle cells are connected to each other for the purposes of being able to have this rhythmic contraction. They want to do it's a different different structure than skeletal muscles. These intercalated discs contain desmosomes, which hold the muscle cells together, but also gap junctions, which allow ions, and it's going to be very important, your calcium, your sodium ions, to pass from one cell to another and be able to communicate with each other. So these cells want to stay in constant communication. These muscle cells, these cardiac muscle cells, want to stay in constant communication with each other. So when the signal comes time to beat, which obviously happens over and over again, that signal gets widespread and they beat together, right? So you'll see the importance of this. This is a diagram just showing you the cardiac muscle cells in more detail, and you can look at this slide, pause, and look at it more closely if you would like. You can see these muscle cells hopefully in section 1 of this class, in Bio 109, Dynamic Physiology 1, you were able to see cardiac muscle cells under a microscope when you did the when you discussed the different tissues in the body. I know there's a section on muscle, and you look at the skeletal muscle, cardiac muscle, smooth muscle. And so hopefully you've seen this slide. You saw this as a slide in part 1 of this class. You can also go back in the book and look at pictures of cardiac muscle fibers as well. All right. Like skeletal muscle fibers, you'll see these gated ion channels in the sarcolemma. You'll see that in cardiac muscle cells. The opening and closing action of these ion channels is responsible for both the pacemaker and contractile cardiac action potentials. And we're going to see this as we saw this in the muscle fibers before. And when you discuss the chapter on muscles, we're going to see how these ions help with the action potentials. And I'm going to say this right now, if you maybe forget how action potentials work, remember, we talked about action potentials, and when we talked about the muscular system, also when we talked about the nervous system, action potentials, you may want to go back and review it. We're going to talk about it here in this chapter. But if it sounds like, wait a minute, I don't remember any of this action potential stuff, you may want to go back and review it. Voltage, the different types of ion channels. You have voltage gated sodium ion channels. So the voltage changes across the membrane. These sodium channels will open or close. And so these are in all cardiac muscle cells except certain pacemaker cells. You also have calcium ion channels. Calcium is going to be very important to your heartbeat. They demonstrate a voltage gated opening, but time gated closing. So they close after a certain period of time, regardless of voltage. And then you have potassium ion channels. Some are ligand gated and some are voltage gated. And you're going to see these different, so sodium, calcium, potassium ion channels as related to the heart. Right, just to revisit some electrophysiology, you're going to see voltage, difference between electrical potential, difference in electrical potential between 2 points. Usually they talk about the outside of a membrane, the inside of a membrane. Membrane potential is the voltage across that membrane. Resting membrane potential, you'll see that R and P. Usually you have to, so it's the membrane potential when a cell is at rest. Usually you have to break through some threshold for it to depolarize. Current is the flow of electrons or ions, so your calcium, your potassium, your sodium, that's going to give you the current. So how these charged ions, these charged potassium, calcium, sodium, these charged ions flowing into and out of the cell, that's what's going to create your current, change the polarization of the cell. Depolarization, depolarized. So the resting membrane potential is normally between -60 and -90 millivolts, as you see here. So your depolarization, becoming less polar, right? So that happens, you go from negative, you start rising closer to 0. You'll see that term depolarization. So changing your resting membrane potential, less negative. And then you have repolarization, as you get back to your resting membrane potential. So going back to your negative 60, negative 90 resting membrane potential. As I said, if all of this sounds like gibberish to you right now, go back and review the chapter on either in the nervous system or the muscular system about the electrophysiology, because that's where it's discussed really in high detail. And those chapters where it's first introduced, and then this sort of builds off that knowledge. All right, so this is the big section. Electrophysiology of cardiac muscle tissue. So these pacemaker cells, which we're going to talk about, undergo this rhythmic, spontaneous depolarization, becoming less negative, that leads to an action potential, right? That action potential spreads quickly through the heart by this cardiac conduction system, which we're going to talk about the 3 parts of that. And that causes the heart, it eventually causes the heart to beat. Action potentials are transmitted from the pacemaker cells to contractile cells through intercalated discs. Remember, those intercalated discs connect the different muscle cells that unite them. These gap junctions allow for electrical activity generated by the pacemaker cells to rapidly spread, right? So spread through the heart, permits the heart to contract as a unit, right? So the heart cells, you have this action potential, these depolarization spreads through these gap junctions that are in the intercalated disc, which connects the different muscles in the these different muscles in the heart, these different cells, muscular cells. This all happens very rapidly, and it allows the heart to be, the muscles to depolarize, the action potential to spread, and for the heart to beat as a unit. So your pacemaker cells make up only about 1% of the total number of cardiac muscle cells. Pacemaker cells make up 1% of your total number of your cardiac muscle cells. That's important to know. And there's 3 populations of these cells, all capable of generating an action potential, and therefore they can, all 3 of them can technically set the pace of the heartbeat. Altogether, this is called the cardiac conduction system. All right, so your pacemaker potential, right? This is, remember, pacemaker cells are your specialized muscle cells that can generate these action potentials, set the pace of the heart. Right? So specialized cells. Notice one thing I want you to notice is it doesn't say 1% of the total number of cardiac nervous cells. It says they're called muscle cells, these pacemaker cells. Just one thing I wanted you to notice. All right, so pacemaker potential, these pacemaker cells that set the pace, set the rate of your heartbeat. Depolarization in pacemaker cells occurs much more slowly due to lack of voltage-gated sodium ion channels in pacemakers, sarcolemma. So these are the differences between pacemaker cells, which are different from that of the contractile cells, the cells that actually do the contracting, do the beating. Pacemaker cell action potentials lack a plateau phase, which we're going to find out that plateau phase is very important in the contractile cells of the heart. So the 1% pacemaker cells, they're different from the 99% of the muscle cells. Remember, this is talking about the myocardium, those muscle cells that are in the center layer of the heart. The plateau phase, which we're going to talk about later, is very important. That allows, so while we have contraction, which is very important because that's when things are contracting, blood is being pushed out, pushed into the systemic circuit, pushed into the pulmonary circuit. The plateau phase is very important because even though we're contracting and relaxing, we have to have a time for blood to actually fill the heart. You know, blood has to get into the atria, blood has to get into the ventricle. So you know, you have to have a break, which is in what we call the plateau phase. You have to have a break in there, just a slight break. You may not have even noticed this, so that blood can you actually have something to pump. The blood has to get in the heart, um, fill up the ventricle or fill up the atria so that it can eventually pump. So pacemaker cells don't have a plateau phase. That's when they're making their potentials. But these contractile cells, the cells that actually do the contracting, have what we call a plateau phase, which is very important. All right, so your pacemaker potential, let's get into that. You have a slow initial depolarization phase, right? So depolarization, slowly going from -60, -90 to 0. It starts with the plasma membrane, hyperpolarized state, at minimum membrane potential. Right, then you have a full depolarization phase when the membrane reaches its threshold. Right, so you get to the threshold, and then you have a full depolarization, so voltage gated, calcium ion channels open, calcium flows into the cells. Then you have a repolarization phase. So it fully depolarizes, then you have a repolarization, calcium ion channels are closed. They close at the same time, voltage gated potassium ion channels begin to open. Potassium ions exit the cell. This helps with the repolarization, so going back to a negative position. And then you have this minimum potential phase. Potassium ion channels remain open until membrane reaches its minimum potential. So becomes hyperpolarized for a moment. And that's just showing you, this is specifically, remember, we're specifically talking about the pacemaker cells, the 1% of specialized muscle cells that set the pace for the rest of the heart. Right? And this is just showing you initially slow depolarization is the threshold, fully depolarizes. Then we have the coming back down, repolarization, and then we have the minimum potential phase. Okay, so that's the pacemaker, just the pacemakers. Talking about the pacemakers. Oops, where did I go? I went the wrong way. All right. Now, the cardiac conduction system includes 3 populations of pacemaker cells. So when we talk about pacemakers, we're talking about the SA, the AV, and the Purkinje. Okay, that's when we're talking. Now remember, all these combined make up only 1% of the muscle cells in the heart. Okay, so with the SA node in the upper right atrium, slightly inferior and lateral opening, slightly inferior and lateral to the opening of the superior vena cava. Right, so the SA node is important because it has the fastest intrinsic rate of depolarization. Usually that's the one that sets the pace for the heart. The AV node, AV, atrioventricular, notice that, posterior medial to the tricuspid valve. Remember the tricuspid, bicuspid, and tricuspid valve. So that's going to be where? Right atria, slower than the SA node, intrinsic rate of only about 40 action potentials per minute. And then you have the Purkinje fiber system, slowest group of pacemaker cells, depolarize about 20 times per minute. And they're a little bit different because their action potentials rely on different ion channels. So they function in a little bit different way, right? But so your cardiac conduction system, the pacemaker cells, the cells that set the pace of your heartbeat, they basically tell the other 99% of the muscles in the heart when to beat. SA node, AV node, and Purkinje fibers. Okay. A little bit more about the Purkinje fiber system, which is a little bit different from the SA and the AV node. You have this atrioventricular bundle, penetrates the heart's fiber skeleton, inferior interatrial septum. You have a right and left bundle branches, and you also have these terminal branches, right? So the Purkinje fiber system more so looks like branches of fibers, right? This is what it looks like in a nice diagram of colors to make it pop. Right, so SA node, that's your fastest one, fastest. AV node, and then you have the Purkinje fiber system, which all this here, all part of the Purkinje fiber system. All right, so notice SA connected to the AV, connected to the Purkinje fibers. Usually, if your heart is functioning properly, the SA node is what is going to basically set the pace for the rest of your heart. But these all work together to get the heart to beat. All right, so pacing the heart, sinus rhythm. So each population of pacemaker cells can potentially pace the heart. So make it beat at a certain pace. Ideally, the SA node is what is going to pace your heart. The one that depolarizes the fastest sets the heart rate. Which one depolarizes the fastest? Remember, go back to this slide. SA node. Fastest intrinsic rate of depolarization, 60 or more times per minute. AV node, 40 or more times per minute. Purkinje fibers, only 20 times, right? So which one's the fastest? 60 times per minute, SA node. And so just, you know, that's something that's very important to remember. So the SA node is a normal pacemaker of entire heart. Electrical rhythms generated and maintained by SA node are known as sinus rhythms. The AV node and Purkinje fibers only conduct action potential generated by the SA node, right? So the SA node sets the pace, sends that action potential. The AV node and the Purkinje fibers further conduct that same action potential. Now, if there's something wrong with your SA node, the AV node can also set the pace of the heart, but you want the SA node to set the heart pace. A note here on this slide, the AV bundle of Purkinje, the AV bundle of Purkinje system is only connection between the AV node and the ventricles. So if blocked, the SA node cannot pace the ventricles. So if there's something blocking if there's something blocking, the SA node can't pace the rest of the heart by itself. It needs this connection. The SA node needs the AV and the Purkinje fibers to be able to pace the entire heart. Also note here, the Purkinje fiber system is capable of pacing the heart, but because it only depolarizes about 20 times per minute, it's not adequate, right? It's not adequate. You can live off of, well, obviously the SA node is the ideal. You can live off the AV node pacing the heart if the SA node was not performing properly, but you cannot live off of just the Purkinje fibers depolarizing the heart because they depolarize so slowly, right? Your heartbeat will be too slow. Remember, your average heart rate is going to be right around 60 to 80 beats per minute, right? And so that's the SA node pacing your heart. So under normal conditions, the SA node generates an action potential, which, like I said, if you don't remember what action potentials are, you can go back and review that in the nervous system and muscular system chapters. It spreads rapidly via gap junctions. Remember, these cardiac muscle cells are connected by intercalated discs, which contain desmosomes, which hold the cells together, but also gap junctions, which allow for the cells to communicate with each other. Right? So impulses are conducted by specialized atrial connecting fibers to the AV node. Once impulses reaches the AV node, conduction slows considerably as a result of low number of gap junctions between the AV nodal cells. You also have a presence of non-conducting fiber skeleton that surrounds the AV node. Right? So your SA node generates its action potential, spreads rapidly via gap junctions. It starts to slow down a little bit once it gets to the AV node. You have this slowdown at the AV node is called the AV node delay. Allows the atria to depolarize before the ventricles, which is what we want. Remember, the atria are connected to the ventricles. We want the blood to go from the atria into the ventricles and not the other way. So allows atria to depolarize before ventricles, gives ventricles time to fill with blood. Also helps the blood from going backwards, which is very important. The action potential is then conducted from the AV bundle to the right and left bundle branches and spreads along the Purkinje fibers. And you can watch the video of this when you get a chance on this slide. Please do, just so you understand the concept. Right, so now let's get into the contractile cells. So we talked about, all of that was basically talking about the cells the pacemaker cells, the cells that set the pace. But we need to get into the contractile cells, the 99% of cardiac muscle cells. Remember, the pacemaker cells are not nervous tissue, they're muscle. The other 99% of your cardiac muscle cells, these are the cells responsible for the heartbeat. The pacemaker cells just set the rhythm, basically, send the action potential, but the contractile cells, these are the cells that actually do the beating. They actually do the heavy lifting, the hard work, so to speak. So what do we have here? We have action potential and contractile cardiac muscle cells results from reversal membrane potential inside the plasma membrane, swings from negative 85 millivolts to a positive 0 to 20 millivolts. So that's your depolarization. Goes from a negative, less polar, to a positive. Voltage-gated ion channels and sarcolemma, unequal concentrations of sodium and potassium. That's what drives the current. You have this plateau phase. Depolarization is sustained, and you have this plateau phase, which is very important, which is due to a slow opening of the calcium ion channels. And then you have this repolarization phase, final phase, which helps you go back to your negative 85 millivolts. All right, so this is the process. All right, so we start off with a rapid depolarization phase. Remember, with the pacemaker cells, there was a slower depolarization phase. With the contractile cells, there's a rapid depolarization phase. So notice that difference. So your pacemaker cell action potentials cause voltage changes in adjacent cells. So your pacemaker cells are causing changes, setting the pace for these contractile cells. Voltage-gated sodium ion channels and sarcolemma are activated. Rapid influx of sodium ions leads to this depolarization. Then you have an initial repolarization, so depolarization. Then you have an initial repolarization, small initial repolarization. Notice that, due to abrupt inactivation of sodium ion channels, small outflow of potassium ions, right? So look at that. You have a rapid depolarization. Notice that line goes straight up, and then you have a small repolarization right here, small repolarization. Then we're going to have what we call the plateau phase. Remember, I talked about we don't have this in the pacemaker cells, but this is very important in the contractile cells, plateau phase, and then we're going to have our repolarization. Okay, so let me see. Sequence of events, resembles that of skeletal muscle fibers. One important exception is plateau phase. So, and if cardiac action potentials only lasted 1 to 5 milliseconds, like skeletal muscle fibers, your heart rate will be 15 times faster. But we have this plateau phase. Not only slows down your heart, which is important, but it also gives time for your heart to fill with blood. All right, that's a major difference between cardiac action potential and skeletal muscles, cardiac muscle and skeletal muscle. All right, so we have this refractory period. So skeletal muscle, you consciously want it to fire, you think, and it fires and then goes back to basically, so you go from your negative to positive back to negative. So you depolarize up and then repolarize. In the heart, you depolarize, very same, have a slight repolarization, but then you have a plateau, and then you fully repolarize. Right? So you have this sort of gap in here, and this is where when your chamber in your heart, the chambers in your heart can fill with blood. It happens fast, but it makes a huge difference. Right? So plateau phase increases strength of the heart's contraction. Prolonged action potential makes muscle twitch last longer. Also, remember, it's very important for allowing the heart to fill up with blood, right? So not only does it allow the heart to fill the blood, it also increases, and it gives the heart a chance to like recoil a little bit and then be able to pump a little bit, pump even harder. So increases the strength of the heart's contraction, okay? Plateau phase also prevents what we call tetany, sustained contractions. That's another issue, but don't worry about that. The refractory period in cardiac muscle cells is so long that cells cannot maintain sustained contraction. So this allows the heart to relax and ventricles to refill with blood before cardiac muscle cells are stimulated to contract again. Very important. All right, so now let's get into this excitation, contraction, coupling. This is the mechanism for cardiac muscle cell contraction. It's very similar to that of skeletal muscles. All right, so it happens by this sliding filament mechanism. Remember, skeletal and cardiac muscle both are striated, both have that very similar structure. In stimulated cardiac muscle cells, depolarization propagates through sarcolemma, dives into cell-long T tubules, causes sarcoplasmic reticulum to release calcium ions. Ions bind troponin. Remember troponin? If you don't remember troponin, go back over the muscle chapter. That allows the actin and myosin to be able to bind to each other, create that cross-bridge cycle. Remember, and it pulls it along. If you remember that, very similar as when we talked about skeletal muscle. The sarcoplasmic reticulum of cardiac muscle cells is much less extensive than in skeletal muscle. Remaining calcium ions needed for the contraction diffuse into cell during action potential. So for this reason, calcium is very important for your cardiac contraction, plays a huge role. Right, so just review that. That's just talk. That previous slide just talking about the method of the contraction, how the muscles, how that sliding mechanism works for the cardiac muscles. Now we get into more the medicine behind, the medical applications behind it, and how we can monitor it, which is probably what's going to be most important to those of you all who are in or who are planning to be in medical careers. Right, so electrocardiogram, ECG. You may have also heard of EKG. But you hear both terms used, but interchangeably. But electrocardiogram, an important clinical tool for examining the health of the heart. Graphic depiction of electrical activity occurring over the course of the heart. So why is this important? As you just saw from these previous slides, we talked about the pacemaker cells, we talked about the contractile cells, we talked about the SA node, the AV node, the Purkinje fibers. We're talking about the mechanism by which our heart beats. And so why is this important to be able to monitor that? If something goes wrong, first of all, we don't have to cut you wide open to see that something else. What we can do, and what we can do even before there is a problem, is monitor the electrical activity of your heart. Because we know how it's supposed to work, we can place electrodes on the surface of your skin. It's actually 6, and this is something that you can actually do. It doesn't hurt somebody to do this to them. If we were in like lab, we could do this to each other. But you place these electrodes on the surface, and you can actually see. You can get a graph of the electrical activity in the heart, and it's really neat to be able to do that because, for instance, I did that, and I'd done it more than once, and my doctor actually noticed at one point that there was a slight issue, which I can talk about a little bit later when we get to what the actual graph means. But you can see that there's an issue. If there's an issue in somebody's heart, like something's maybe not happening, some part of your heart is not firing like it's supposed to, there's a delay or something not quite right. You can see that right away. All right, so we can see the waves of the electrical activity. We can also see if there's any kind of issue, dysrhythmia or arrhythmia. We can see like the different disturbances, and we're going to talk about what those differences are. So the ECG, electrocardiogram, generally consists of 5 waves, represents active depolarization, that's going from negative to positive, or repolarization, going from positive back to its negative resting membrane potential, different parts of the heart. So you have a small initial P wave, which represents the depolarization of all cells within the atria, except the SA node. So depolarization, going from negative to positive of the atria. That's your P wave. Always registers as an upward deflection, so from negative to positive. Then we have what we call the QRX complex, which represents ventricular depolarization. Depolarization, negative to positive. Actually, 3 separate waves. Q wave is first, downward deflection. R is a large upward deflection, and S is a following downward deflection. Right, we'll talk about that. It's the ventricular depolarization. And then we have the T wave. It occurs after the S wave of the QRS complex, represents ventricular repolarization. T wave is upward deflection under normal conditions. Repolarization going from positive to back to negative, back to its negative resting membrane potential. Right, so here we go. The atria depolarize, P wave. QRS complex, ventricular depolarization, slightly down, then goes up and back down. So that's your ventricles depolarizing. And then you have what we call ventricular repolarization in your T. Periods between waves represent important phases of action potential. Intervals include component of at least one wave. Segments do not include any wave components. So these are just terms we use to describe the different parts of the ECG. All right, so P to R, time between two successive R waves, entire duration of generation and spread of action potential through the heart can be measured to determine your heart rate. That's R to R. I apologize, R to R, right? Going from successive R waves. Remember the R is so that's your point of ventricular depolarization. So if we go from one apex, one R, to the next R, that's basically the length of your heartbeat. P to R interval, period of time beginning from beginning of P wave to beginning of R wave, time it takes for depolarization generated by the SA node to spread to the atria and ventricles. So let's go back. P, that's your depolarization of your atria. How long does it take to get from the depolarization of your atria to the depolarization of your ventricles, which is your R? That's your P to R interval. And then QT, time from beginning of QRS complex to the end of T wave. So that's your action potential spreading through your ventricles, right? So Q, that's where your ventricles, that's where ventricle depolarization starts. T is where your ventricular repolarization. So from Q to T is the action potential spreading through your ventricles. P to R, action potential atria, from the atria to the ventricles. Q to T, spreading through the ventricles. Okay. And these are some these are those diagrams also as well. So R to R, entire duration of a cardiac action potential, or your heartbeat. Do-do, 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 beep, beep. P to R, atrial depolarization. Q to T, ventricle action potential. You also have what we call ST segment between the end of the S wave and beginning of a T wave. That's your plateau phase of ventricles. Elevation or depression of the ST segment is seen with many clinical conditions, most notably myocardial ischemia or myocardial injury or infarction, heart attack, right? So it should be flat because it's a plateau phase. But if there's an issue, then there's an elevation or depression of the ST segment. Let's see, right. Picture is blocking it. Let me move that. So that's your ventricular plateau phase. Why is that important? Plateau, filling of the ventricles. But if there's an issue, which it doesn't have it in this diagram, we don't have a diagram of that, it can be because there is an injury to your heart. That's your ST segment. Okay. This should generally be flat. All right, so this is just another diagram showing you those different intervals. The plateau phase, QRS complex, depolarization of your ventricles, P to R, atria, Q to T, ventricle depolarization and repolarization, the action potential spreading through the ventricles, P to R, action potential spreading through the atria to the ventricles, R to R, heartbeat, heart rate, right? So know all those terms. And you can have what we call dysrhythmias. We mentioned that a little bit earlier. All right, so your cardiac dysrhythmias have three basic patterns. Bradycardia, heart rate under 60 beats per minute. Remember, your SA node fires at about 60 times per minute. That doesn't mean your heart rate is going to be 60 times per minute always. Obviously, your heart can beat faster. Your bradycardia is under 60 beats per minute, and that can happen for different reasons. It could be because you have a really healthy heart and your heart doesn't have to beat as fast when you're resting. So that you'll find like people like long distance runners, a lot of times their resting heart rate can be as low as 50, 40 beats per minute. And that's considered bradycardia, but it's not considered unhealthy. Um, there are other reasons that it can happen that are not good, obviously. Tachycardia, your heart rate is over 100 beats per minute. Um, Usually children, when they're first born, their heart rate is faster. That's something that's a natural way that it can be a little bit faster than normal. But if you have issues with your heart will have to beat faster. Also, if you are um, exercising, if you're nervous, you'll notice your heartbeat can elevate very quickly. Sinus tachycardia is considered um, regular fast beat. All right, so these are things that are very important. Your heart beating too fast or too slow can 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 be due to specific issues, and so we need to investigate if either one of those is the case. There can be disturbances in the conduction pathway that can cause problems. And we talked about the pacemaker cells, the SA node, the AV node, the Purkinje fibers. And so you can have blockages at the AV node. Now the PR interval is longer than normal due to increased time for the impulse to spread to the ventricles. Extra P waves present indicate some action potentials from the SA node are not being conducted through the AV node at all. You can have issues that way in the conduction pathway. These are all things that we can basically get from their ECG, EKG, whichever one you want to call it. AV node blockage. Another common location for heart blocks is along the right or left bundle branch. Generally widens your QRS complex. Depolarization takes longer to spread through the ventricles. Longer time for depolarization. It's going to be slower heartbeat, bradycardia. We can also have in fibrillation, electrical activity in heart essentially goes haywire. This is not good. Not good. Causes parts of the heart to depolarize, contract, while others are repolarizing. Remember, there's a reason why your heart beats the way it does. You want the blood to go in a certain direction. You want it to fill the atria, then go from the atria to the ventricles, and then go from the ventricles to the pulmonary system, pulmonary circuit, or the systemic circuit. Right. So if it starts beating uncontrollably, it's called fibrillation. Parts are depolarizing, repolarizing, and the rhythm is off. Obviously, the blood flow is going to be off, and that can cause problems. Fibrillating muscle is often visually compared to writhing movement, Plaster-Bath run. So, like it's uncontrolled. It's not. The heart's not in rhythm. That's not good. So, atrial fibrillation is generally not life-threatening, because atrial contraction is not necessary for the ventricle filling. So the blood can go from the atria to the ventricles more so just by gravity. It can go, it doesn't necessarily have to be pumped in that direction. Remember, the atria just separated by the ventricles from just a valve, right? So notice here manifests on the ECG. ECG as irregular rhythm, irregular rhythm that lacks P waves. Notice the P wave is missing. That where the atria depolarized, that P wave is missing. So that's how we know we have some atrial fibrillation. We have the QRS, so we have the depolarizing of the ventricles, and we have a T, so we have some repolarizing of the ventricles, but we're lacking the P wave, and that's how we know that we're in atrial fibrillation. The atria aren't firing exactly when they're supposed to fire. They're firing sort of out of control. Ventricle fibrillation, that's problematic. Remember, the ventricles send the blood to the systemic and the pulmonary circuits. And if they're not firing when they're supposed to, remember, they need to fill up with blood and then push that blood through their respective circuits. Now we have trouble. And so here we have, it's something that's immediately life-threatening. Your blood is not going to get to your lungs. Your blood is not going to get to the rest of the body like it's supposed to. All right, so this is treated with defibrillation, right? So fibrillation, things aren't beating on rhythm. So we have defibrillation, which is electric shock zoop, to the heart. Clear. Y'all, I'm sure you have seen that on TV. The idea of that electric shock is to depolarize all the ventricular muscle cells, right? So try to get all the cells to depolarize, go from negative to positive, depolarize at the same time, and hopefully throw them into that refractory period, that plateau, that refractory period. And then the idea is you want them all to come back, repolarize, come back, and start beating together. That's the idea. Ideally, the SA node resume pacing the heart after the shock is delivered. One cause of ventricular fibrillation is scar tissue on the heart after a heart attack. Like I said, having a heart attack is bad on the body, bad on the heart. You know, bypass surgery, stents, balloons, potential for ventricular fibrillation. It's good to take care of your heart. I just put it like that, very simply. So if you got fried chicken on the menu today, take it off. Or just eat a little bit. This slide is just saying that ventricular fibrillation is not the same as flatlining, which is asystole. Flatlining is when obviously there's no, from the ECG, EKG, flatlining is when we have nothing. No repolarization, no depolarization, no P, no QRS, no T. This is treated with CPR. This is when you need cardiopulmonary resuscitation. Your heart's not beating, right? This is why we get into, this is when we get into use of CPR. With ventricular fibrillation, we can shock the heart and try to get it back into a regular beat. When it stops beating, we need to get into CPR immediately. Also, you can treat it with epinephrine. There's other ways to treat it as well. Okay.